Hello students, welcome back to my class. So dear students, in my last class I had discussed you about incomplete dominance, right? So in today's class I shall be discussing about codominance. So let us try to understand what is codominance. So till now you have learned about the dominant factor, recessive factor and incomplete dominance, right? So let us see what happens in codominance. Generally what we learned is that in F1 generation, in the first we learned that the dominant character overpower the recessive character, right? Then in incomplete dominance we learned that the dominant character doesn't mask over the recessive character completely, and as a result the F1 progeny, the result came something different, right? So now let us see about the codominance. So in codominance, what happened here? Both the parents express their character. Okay, so the F1 generation, here the F1 generation, they resemble both the parents. Here both of them equally. Okay, both the parents express their character equally. So the best example for codominance is RBC, that is your red blood cells in human being, and this red blood cells, it determine the ABO blood group. See, we always whenever we go for a test or we want to know that the blood group, right, that some has A positive, some has B, some has AB, some have O blood group, right? So that is all this RBC determines. So ABO blood group, and this ABO blood group is controlled by gene. See, ABO blood group is a kind of trait, right? It is a kind of character. So this is controlled by gene, and that gene is your I, gene I. So ABO blood group is controlled by gene I. Now the plasma membrane of RBC. See, RBC means red blood cells. So cells, they have plasma membrane, right? So plasma membrane of RBC, they have some sugar polymers. Okay, they have sugar polymers, and the kind of sugar, okay, the polymers, the sugar polymers is controlled by this gene I. See, what you have to understand here is gene is controlling the thing. Okay, gene is very important, that carrying the character, and it is the main factor, it is the important factor, and it controls everything. Okay, so similarly this ABO blood group is also controlled by gene. Now this gene has three type of allele, that is your IA, IB and I. Correct? So three types of allele are here. Now out of these three alleles, IA and IB are dominant. They are dominant because the sugar produced by them, okay, IA, gene I and gene IB, both of them, they produce sugar, and the sugar produced them by them is slightly different from each other. Okay, and they are dominant also. On the other hand, the lowercase I, the small letter I, okay, it does not produce any sugar, therefore it is known as recessive gene. Okay, it is known as recessive. So remember that gene IA and IB, they produce sugar, and they are slightly different from each other, and allele I, it does not produce any sugar, therefore it is recessive by nature. Now let us see how this gene expressed in human being, or how does it express the blood group. Okay, I hope with this you will be more clear about the codominance. So say for an example, parent 1 and parent 2, okay, and the progeny. If a parent, if his blood group is A, okay, A means either it has to be IA or I. I hope you all can follow this. And on the other hand, suppose parent 2 blood group is B, okay, so the alleles present here will be either this or this. Now here you see, this is blood group A, and this is this allele also for A, right? Why? Because A is dominant over this I, the lowercase I. Now the blood group that progeny can have, okay, there are some possibilities. Okay, let me show you here. So first what happens, suppose let us take this one. So from here one gamete will come down, right? And suppose the first one here 1, then what happened, the blood group will be your IA IB, that is your AB. Now why AB? Because as I said that here both of them they produce sugar, and these are slightly different from each other. Okay, and both the parents show their characters in F1 progeny. So this is your F1 progeny, right? So in F1 progeny both the parents are expressing their character. Am I clear? I hope that you all are clear with this. Okay, now let us see. Suppose, say for an example, for another alleles for this. So here two gametes will form. Okay, same with this. Now the possibilities of blood group. So sorry. So the possibilities of blood group is either it could be this. Okay, it could be this. It could be this or this. Okay, so here you see, if it is this, then it will be your AB. If from here, if IA comes and from here the small letter I, if the recessive alleles come, then the blood group will be your A. Why? Because A is dominant over this. And if from here, if recessive alleles comes and from here the dominant allele IB comes, then the blood group will be B. And if from both, if both the recessive alleles come, then the blood group will be your O. Am I clear? So this is how the blood groups, okay, are determined based on the alleles, the dominant allele and the recessive allele. Am I clear? So these are the possibilities. So this could be reversed also. If the parent one is blood group B, okay, the allele could be this, and if it is blood group A, so the allele will get reversed, okay. So the progeny will be this. So this is the possibilities. Am I clear? Okay, let me show you some more example. Suppose both the parents, okay, one of the parent possesses this, okay, IA, IA. And here, suppose it is IA and I. So from here, one gamete will come, and from here, two, because both are different, right? So the possibilities is either this or this. But in both cases, the blood group will be A. So this is called your codominance. I hope that codominance is clear to you. Just remember that co means both, right? So both the parents, they express their character. And in F1 generation, the both parents resembles. Okay. So I hope that this is clear. So next we will learn. Okay. So now let us try to understand inheritance of two gene. So previously I had taught you inheritance of one gene, where the Mendel has chosen only one character, right? That is your height, isn't it? So where tall and dwarf character were chosen and the experiment was carried out. So what happened? Mendel tried this experiment with two gene as well, means with two different characters. So the character he had chose was your round yellow, where both of them were the dominant, okay, round shape and color. Okay, so what he did, he took two traits together, that is the color and the shape of the pod. So here the round is dominant and yellow is dominant, okay, and here you can see the wrinkle, okay, wrinkle green, both of them are recessive character, correct? Okay, so the genotype symbol for round is capital R. Okay, I hope you all can follow. So this is capital R and this is capital Y. It is your round yellow, and this is your wrinkle gene, small letter R and Y. So from here, the R, one gamete came from here and this. So RY, and here also RY, the recessive allele. Then the progeny, the F1 generation is your capital R, small r, capital Y, small y. So according to the Mandel, the first generation is what? Always dominant, isn't it? So out of this, round is dominant, right? And yellow is dominant. So round yellow. So the F1 progeny was round yellow. So then what he did self-pollination, selfing of this. So in that way, he got this allele. So how it is done? So this say RY, selfing with self, right? RY, then capital R, small y, R, Y, and here both are recessive. Now after selfing, let us see what are the results that he found, okay? So this will be your round yellow. Then comes your this. Again round yellow. Then RR and capital Y. Round yellow. Then round yellow. Then here you see the second one is again round yellow. I hope you all can follow me. Then comes your, okay, here you see this both a small letter. It means it is green, right? But the shape is dominant, round green. So this is something different, okay? Then comes your, again, round yellow. Then round green. Then, I hope you all can see this. Then comes your round yellow. Then again round yellow. And here wrinkle. See, both came small letter. Okay, I hope you all can see. And yellow, right? So this will be your wrinkle yellow. See what we saw here, that dominant character came together, right? But here when the selfing is done, you see the variation. So we will discuss this, okay? Let us see what else comes in the result. So R, Y. So this will be your wrinkle yellow. Wrinkled yellow. And here is again round yellow. Then round green, wrinkle yellow, and wrinkle green. See, this is your last one is your wrinkle green. I hope you all can see this. Okay. So, what we got from here is round yellow is to round green is to wrinkle yellow is to wrinkle green. 9 is to 3 is to 3 is to 1. Okay. Now see, what does it infer? See, I hope you all can follow. This is round yellow, this is a dominant character, and this is a recessive character, right? In first generation, dominant character mask over the recessive character. But when this selfing is done, okay, after selfing, what we found is round yellow, means the dominant, pure dominant character we found is 9 out of 16. Then round green, where round is dominant and green is recessive. So we found round green 3. And then we found wrinkle. Wrinkle is recessive. See, recessive character. And yellow 3. Yellow is dominant character. And then we found wrinkle green, that one of the parents. See, round yellow is the dominant parent. We found 9. This is the intermediate, okay, where both the character or both the alleles from both the parents came. And in this case, we found only one homozygous recessive trait. Okay. Now, what does it infer is that during the segregation, okay, when they segregate, when the gametes segregate, okay, this gamete, they are independent of each other. Though they remain together, but when they get segregated, they doesn't depend on each other. They get separated, they become independent. Means here you see, here the all segregation is done, right? They were not dependent. That's why the different character blend with the different, right? Here you can find the recessive and dominant character as well, isn't it? So here they just came together, right? Because they were not dependent on each other. So from this experiment, the Mendel has given one law. He proposed one law based on this based on this experiment, and that is known as your law of independent assortment. And it states that during segregation, okay, the gene, they doesn't depend on each other, they are independent of each other. Okay, so let me give you the proper definition of this. So here you can see the page number 80. Okay, in page number 80 it is said that paragraph 2, okay, and reading out this paragraph, it is said that based on upon such observation on dihybrid cross. See, this is called your dihybrid cross. The first one, where we have found law of dominance, law of segregation, that was monohybrid because only one character was there. Remember that this is your dihybrid cross because two different characters are taken for this, okay, dihybrid cross. So the dihybrid cross, so crosses between the plants differing in two traits is called your dihybrid cross. Mendel proposed second set of generalization that we call Mendel's law of independent assortment. So from here, second law, the third law, sorry, is proposed. That is called your Mendel's law of independent assortment. And this law states that when two pairs of trait are combined in a hybrid, see what happened? Two pairs of trait, right? Combined in a hybrid. Here you can see two pairs of trait are combined. This is one pair, this is one pair, right? Combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. So here you see when they get segregate, okay, when this pair gets segregate from this, they are independent of each other. So it doesn't mean that the characters, their traits depend on this. They're independent of this allele, this trait. So it means that they are both of this allele, the pair of allele are independent of each other, and this is called your law of independent assortment. And this cross is called your dihybrid cross. And here is the phenotypic ratio. Here we get round yellow where both are dominant, the 9, round green 3, wrinkle yellow 3, and wrinkle green, the pure recessive 1. So I hope that inheritance of two gene is clear to you, and still if you have any doubts then let me know. Okay, I'll try to solve it out, I'll try to clear it out. So this is all for today's class. I'll see you in my next class. Till then, write down notes on this topic. Okay, so I'll see you in my next class.